So today I'd like to talk more about gene to GMP in 9 months. So how can we reach the speed to the clinics, and how can we accelerate the number of drugs that goes into the clinics? So I'm going to be talking both Celexis and KBI, and I'll know more obviously about Celexis, but I know some about KBI. So basically I want to walk you through this agenda with a history of our collaboration, then the key strengths of both companies, some data, and then timelines. How do we reach 9 months to GMP? And then the non-compromise on quality, because quality is important. And finally, do we believe we can do that in 6 months? So our collaboration with KBI started already back in 2012, when KBI developed a bispecific antibodies process. And I don't know if you remember, but the biospecific is complex to make, because on the cell lines you need to generate a cell line that produces the heterodimer. And KBI had to develop a process by which they were able to purify that heterodimer. And actually they were very innovative in the downstream process, and we were very successful. And in 2015, KBI was acquired by GSR Corporation, a Japanese company that wanted to invest in life science. So basically what GSR acquired was innovation. How can we innovate in manufacturing, up and downstream, including analytics? And in 2017, actually, during the last BIO, we announced that GSR Corporation acquired Celexis. And their idea was we want to put even more innovation upstream in the cell line making. And it took us a year to figure out how we could launch a new initiative where we basically work from the genes, from the transfection, to the bulk process, or actually the bulk substance, in 9 months. And it took us a year to make it, and at BIO a year later we can announce it. And the idea is really, can we bring innovation to the CDMO industry and ensure that we can have even faster process while keeping the quality intact? So fastest time means faster to the market. The first to the clinic will be the first to the market. Clinical stage is uncompressible, so you're the first on the market. So what we want to do for our partners is, can we help them first to get drugs to the clinic? And one of the critical stage is, which drug am I going to bring to the clinics? And we see more and more of our partners that come to us and they want to assess many different genes in order to have the key points, the decision point to bring that drug to the clinic. And of course, if we do so, then we have the best expressing cell, the best manufacturing platform, therefore we should act on the cost of goods. On Celexis side, what have we done in the past 10 years? We have 100 clinical programs that actually use Celexis cell lines to produce the drugs. We have 3 commercial products. We have 154 granted patents. And over the last 10 years, we've launched around 9 new key technologies, not only on the cell lines, but also responding to new challenges such as difficult to express proteins. On the KBI side, KBI is unique because they develop over 100 process development programs or manufacturing batches. They file 12 INDs per year, which is amazing. And I think what is most important on the KBI side is they've done 240 analytical service projects. And it's an amazing amount of data and an amazing amount of know-how that is important. And together on the G2G, gene to GMP, what we've collaborated is basically we've achieved programs, MABs, molecular antibodies and non-antibodies, achieving titers of about 2 to 10 gram per liters. And since 5 years, we've collaborated on 10, actually KBI collaborated on 10 Celexis cell line. So we've done some work together before even the acquisition happened. So now on the G2G, what is specific for each of the partners, Celexis and KBI? Well, at Celexis, what we've done is we have 4 pillars. One pillar consists in how do we construct a gene expression vector so that that gene expression vector, once integrated in the genome of the cell, is actually transcribed, translated, and secreted. We've worked that out, and that's what we call the SHOW technology. And basically, we base this approach on epigenetics. Epigenetic is a piece of DNA that actually modifies the structure of the chromatin, therefore it's an open state. It's transcriptionally active, so if you have more transcription, more translation, and then more secretion. So that's what we do. We also have a good choke K1 cell, okay? I will allude to that a bit later, but it's so important. You know, bringing a product to the clinics and to the market is building a house. You need to have the foundation rock solid, and that foundation starts with the cell line. Then, of course, what we have to have with the cell lines is you need to have a media. So we have off-the-shelf media, we have off-the-shelf feeds, and we also transfer strategies. And with KBI, we've established strategies in order to have actually a smooth transfer from the RCB to the process. And more recently, we went into launching a new initiative which we call the whole genome sequencing. It's basically the idea behind it is we sequence the genome of our CHO, we sequence the transcriptome of our CHO, so now we can fully document them. And one of the immediate impact is that by having the sequence annotated of our CHO, we can now certify and authenticate all our research cell banks. So we provide a regulatory package to the FDA, and actually we're discussing with them on how to package that more efficiently. And to me, the ultimate goal is, can we barcode cells? Because you need this barcode because that cell will be used for the life of your drug. So you have to ensure that your starting point, the foundation, you have a clear idea of what the foundations are. So I think that's where is the strength. And that's a marketing slide. Celexis KBI integrated workflow. So we're based in Switzerland. We like ski. And actually, you know, when you do ski, you need speed, precision, strategy, agility, and confidence. But actually what we built in with this integrated workflow is really how can we speed up the process. And on Celexis part, it's pretty simple. We have a DNA. We need to transfect CHO cells with that DNA and ensure that the DNA goes into the nucleus, is integrated, and then we get to a cell pool. And at that stage, we have the first interaction with KBI that starts. So that's why we can be rapid up. Try to sort out the upstream process rapidly enough so that once we reach the research cell bank and then the MCB, the process is almost done, and then can work the downstream process on it. From the KBI side, KBI is a world leader in analytics, and that's not disputable. And they've been the leaders in their ability to really look at the product, a biological product. What they added after that was they added this analytics capacity to the process. Can we use analytics and look at the process? And they've been always innovative. I think KBI was the first company to implement the Amber bioreactors in the world, and now everybody uses Ambers. And you use Amber to choose which of the 2 or 3 clonal cell lines you have that has the best behavior for the next bioreactor. And they really take on this leadership. And what they did afterwards, they were the first one to actually use 2,000 liters bioreactors disposable. And that was a change, you know, from stainless steel to disposable that was imposed by KBI. And finally, they are inspected by all the regulatory bodies, so they can release drug now, and they build commercial manufacturing capacity so that we can respond actually to the demand. And I think, as I said before, they've done over 240 different molecules. They've analyzed them. So that means it's a huge amount of information they accumulated, it's data and it's know-how. So it's much easier when you get there to have those data back when you analyze one drug. They have 14 years of leadership in formulation, and formulation starts already when Celexis transfers supernatants very early on. So you can initiate formulation process so that when the MCB is done, you can formulate your product. And finally, they're always aware of new technologies such as analytical ultracentrifugation or new product characterization using, for example, cell-based assays. So you have all these packages that allows you to really analyze the product. And then you go in the process. I won't go too much into details. You can go to the booth of KBI to get the details. But the objective is, once we have the research cell bank, it goes to master cell bank. While you do that work, you can already work on your upstream and see your different strategies so that when the MCB is released, you can immediately plug this MCB into your bioreactors and go up to 500 and 2,000 liters and get your bulk drug substance purified. So that's basically the objective. You know, how can we really work as close as possible and as fast as possible. So now, if I look at the programs we had, if you look at biospecifics and at FC fusions, we had programs up to above 6 grams per liter. And again, if you look at a biospecific, it's actually 3 or 4 different heavy and light chains, genes. So what happens is not only you have to do a cell that produces more than 90% of the heterodimer, but then you have to have a process in place to purify this heterodimer to almost 100%. And that's really remarkable. Now, on the antibody side, we've done up to 10 grams per liter. And I need to say something here. Everybody will tell you, oh, if it's an antibody, it's 10 grams per liter, easy. But actually, it's not easy. And we have antibody programs that do not do more than a gram per liter. And they are not all easy. So we should forget of always thinking it's easy to make a product like a biologics. No, it's not easy. Antibodies are slightly easier than other scaffolds, yes, because they're naturally expressed by B cells. But other than that, they are not always easy to express. So if somebody tells you, you should get to 10 grams, it's sometimes more complicated than that. So basically, the timelines we're achieving is basically 9 months. When we start the transfection, the clock starts ticking, and 9 months later, we have the bulk drug substance purified that is ready to be filled and finished and actually injected into patients. So we have no compromise on analytics, as I said. You have the analytics on 2 sides, on the DNA side and on the product side. On the DNA, we launched this whole genome sequencing. Celexis is still the only company that is able to extract the genomic DNA and to sequence it using next-generation sequencing. And everybody will tell you next-generation sequencing, that's cheap, easy. Yes, it's cheap, it's easy. But the data analysis and the bioinformatics is extremely complicated. And that's where we bring something on top of it, Celexis. So we looked at clonality. We looked at purity. We looked at transgene integrity. Is the gene that is producing my product the right sequence? Is the sequence that I patent protected intact? Where is it integrated? The site of integration is extremely important. The gene survey, what we want, in a CHO genome, some portion of these genomes are haploid. So imagine if you have a mutation that ends up to kill this haploid gene, then you may have an issue in your process. The cell may die. So how can you de-risk and anticipate such a mutation so that you can stop and start it again? I think that's extremely important. And of course, adventitious agents, same story. What you have to do is the viral content of your cell. So what happens is if that change or you have an increased viral sequences in your system, you had a contamination. You stop your process, clean, and start it all over again. So I think that's what we call the barcoding system. And at the DNA level, you can do any transgene and you can barcode it. Now on the drug substance, what you can do is look at titers, aggregation, purity, charge variance, glycan structure, binding potency or biological potency, and antigenicity. And that KBI has done on almost any biological substance. So truly, not only we speed up the process from transfection to the bulk drug substance, but we bring in the analytics so that we ensure that our partners are going to go with the right substance to the patient. Now, can we do it faster? So can we even improve the speed at which we generate those? I said 9 months to GMP from transfection. I think we can do it in less. So how could we do it in 6 to 8 months or 6 to 7 months? I think we need innovation in the making of the research cell bank. And I believe that at Celexis we'll be able to actually release a new process by which we generate a research cell bank in 9 weeks. Okay? We're also working on improving our host cell. Not only we sequence the genome, we have the transcriptome, so we can improve the metabolic content of that cell, so we can improve the stages of the cell. But we're also working on trying to remove all the viral sequence the cell is secreting. If you have a process, you secrete these non-infectious viral particles. So if we create a CHO cell line that is devoid of those viral particles, maybe one day in downstream we'll avoid one of the viral clearance steps. So it's basically, can we plug in as much of the upstream development at the RCB stage so that it's a lean process to production. So again, I think if we line up OSD, will be maybe next year at BIO, I'll be talking about 6 months to GMP. It's a challenge. So now I'd like to conclude on saying a few words on it takes a village to make a map. Why? Because I think on the drug discovery part, you have to think that it takes at least 3 to 5 years to get an antibody against the target, once you validated identify and validate the target. So the true value point of your product will start the day you've decided which one goes to the clinic. That's a value triggering point. And of course, when you get there, the hurdles are all of there. What is the COGS? What will be the success rate? And all of the hurdles. This is why we see more and more companies coming to Selexis, trying to look at many different variations of their product and try to link expression potential in CHO to those numbers. And then, of course, when we talk about CHO, how fast can we get there? Can we avoid amplification step, whether you use methotrexate DHFR or use the GS system from Lonza? They all amplify and duplicate genes. And you're creating stability and difficulty in isolating high-producing cell clones. So basically, it's important. And most of our partners, they look at, don't ask me why, 1 gram per liter. The go, no-go decision is 1 gram per liter. And again, it's not that easy to get 1 gram per liter cell line. It's not that